Good morning. A very good morning to all of you. This is Dr. Saikat Malik working as an assistant manager from Anchrome. On behalf of my entire Anchrome team, I welcome you and our guest lecturer for today for this another session on a different topic of today. This has been a continuous sessions which have been taken by Anchrome, and thanks to all our guest speakers who have been so kind to provide their support for these sessions and also helping young research minds, industry, government sector, all the scientists from all fields. They have been drastically benefited from these sessions and continuously being benefited, and we look forward that they have the maximum knowledge from all our associates. Our today's guest lecturer is very special. He has been a very long since known person for Anchrome, and since then, as a researcher, as a scientist, has been a very dynamic person. He has published several papers, also come in news channels for various activities which he has performed in his facility. And so it really gives us immense pleasure and honor to introduce our guest lecturer, Dr. Kashyap Thummar, who has been long been associated with Anchrome. For those who are new to the session, I will introduce you that he has been currently working as an assistant professor at Graduate School of Pharmacy, Gujarat Technical University, which is one of the pioneer universities in India, doing a very drastic research work in the field of sciences. He has almost given 11 years of teaching and research experience. He has joined the Department of Pharmaceutical Sciences, Saurashtra University, as an assistant for professor in 2011. His research work of PhD was recognized by University's Grant Commission UGC and received a prestigious UGC Basic Research BSR Meritorious Fellowship for 2013 and 18. So that really defines him, that how dynamic he has been through his career, and is still continuing and pursuing it, and completed his PhD in 2018. Since 2018, he is working as at Gujarat Technical University. His research area focused on stability testing, impurity profiling, bioanalysis of drugs such as therapeutic drug monitoring, pharmacokinetic studies, and human biomonitoring of hazardous compound. He has guided a large number of master students. He has the expertise to handle several analytical instruments like HPLC, HPTLC, LC-MS, Headspace, GC-MS, MS, Preparative HPLC, AAS, FTIR, and many others. He has completed many research projects on a consultancy basis from the academia as well as industry project from all over India. He has received various grants from Gujarat Coast, UGC, SSIP, AICT, DST, SERB, and others for seminar, training program, research project, and also research international travel grant. He has published 17 research papers in national and international journals, 2 patents on HPTLC method, and 1 book from German publication house in his credit. He has given an expert talk at several national and international conferences, and this summarizes the entire gist of Sir's dynamic personality. Although this is a very short gist of what he is, but technically we have been known to him, and he has been absolutely non-biased towards science. He has justified all dynamic fields for research work, and whenever any requirement was or problem regarding any analysis came up, he gave up dedicated support to it. So with this kind note, excuse me, sir, if I have missed any points in your introduction, but this is our today's lecturer, Dr. Kashyap Thummar. Once again, on behalf of entire Anchrome team and our respected researchers and delegates who are there in this session, I welcome you, and we look forward to have a good topic discussion, which is very different from normally which we have. He has taken a unique topic for today, which will be really giving us insight of the entire process in this journey. So with this note, sir, I request you to take over the session and proceed with the presentation for the insight. Thank you so much, all of you. Before sir starts his lecture, I would like to highlight that we have a Q&A box at the bottom. Whatever questions you wish to ask him or us, you can write it over there. He will take up few questions in the session, and after the session, those questions which are not answered, we will be replying those through email. So don't worry about that. With this note, I hand over the session to Dr. Sir, Dr. Kashyap ji. Please, sir, I request you to start the session. Thank you, sir.
Namaste all and good morning to all. Very much thank you, Dr. Saikat ji, for inviting me. I am also thankful to Encrom team who have invited me for this forum to discuss something new about HPTLC. So I am very much thankful to all the team of Encrom. Okay. So looking to this topic, HPTLC a good choice for extractable leachable study. Generally, HPTLC, what we think that HPTLC is suitable for herbal analysis or any kind of things, but the technique has some potential which can be also applied to this kind of areas like extractable and leachables. So today we are going to discuss my 2 to 3 years of work on HPTLC. So before starting my topic, I will just highlight about my university. There are just 2 to 3 slides of my university, okay, where I am there, because those who are because this presentation is going to be all over India. So I'm just would like to highlight that regarding my university. So this is an outline of my presentation. So I will highlight about my universities and the 2 topics that I have mentioned in title, like one of extractable leachable from pharmaceutical product, and another thing is the biomonitoring of extractable leachable, and third one is the comparison with the methods of with LC-MS. Okay, so we are Gujarat Technological University. It is a state university and more than 400 affiliated colleges and more than 4 lakh students are currently there. The GTU is currently main domain is engineering, pharmacy, architect, management, and computer science. So those who have in Gujarat, those colleges from the engineering or this background, they can be affiliated to GTU. These are the few highlights of our department, the timeline, Graduate School of Pharmacy. Our department is granting a department and it is just started in 2017. It is just 3 or 4 year old department. We have started MPharm courses. This is exclusive PG department. Our lab are also approved as per the public testing laboratory from FDCA Gujarat. And we are now in a very much development stage. So we have exclusive PG department as well as public testing laboratory. So our students are having experience to get the knowledge of public testing, how to work in the regulated laboratory. These are the few laboratory facilities. We have almost starting from analytical perspective, unknown to known. We have all the instruments like UV, starting from UV to NMR. Okay, we have all the facilities in our laboratory setups, and in HPTLC, we have a complete range of the accessories or assemblies that is shown here, like starting from sample applicator to automated development chamber, deratizer, photo documentation, scanner, and HPTLC mass interface as well. So now coming to the topic. So as I said, HPTLC very well known for the herbal analysis, or we can further normalize that. Wherever the complex matrix is there, HPTLC is find suitable, or rather than the other techniques. So in extractable leachables are also same things. The complex matrix is there. So I thought that why cannot we try to opt this technique? So means we start from the introductions. Those who have a very new words for the extractable leachables. So generally, drug container and modern drug delivery system, these are made up of plastics. Okay, and plastics have used means that the container is protect the drug from the environmental contaminations. but sometimes they are actually themselves as a source of contaminations, because we are using the plastics, and for making of plastics we are using the uh, many things like plasticizers and to make a durable, flexible. So that are freely there, and it can be leached to the product. Okay, so if what is the extractable leachable? If we test the material, and the material that will be coming to the market, that is a extractable. Leachable means test the product, means from product we are measuring the whatever the content is there from the container or not, that is called as a leachable. So for any organic or inorganic chemical entities that can be released from the packaging into the product, that can be called as a extractable or leachable. So it is there, extractable and leachable, it is there, means in any, it is not about the any pharmaceutical product. Suppose we the milk is also packed within a plastic product. Water is also packed in plastic bottle. So wherever plastic is used, it is more chances to have a the material which is made for the plastics that will be released into the product, and that product is continuously taken by us. Okay, and it have a very severe side effect. So we have also discussed about this. So why worry about ELS? Okay, extractable leachable, why we have to worry? First of all, if the extractable and leachables are found in products, so they will affect efficacy, safety, and quality of the pharmaceutical product. So you can see in 2014, FDA drug recall surged over 836 in 2014 due to safety and quality issue. So it is due to the product recall by the safety and quality issue. And then in European Union, it is third most significant category for product recall, which is related to the packaging material. So it is again uh, the the product recall, and this is the third most significant category for the packaging issue, the packaging issue related issue, and due to this, that will affect the safety and quality, and it is the third most significant category in the European Union. So what are the harmful compounds identified as extractable leachables? So for making of plastics, several compounds are used like phthalates, toxic elements, polyaromatic hydrocarbons, vulcanizing agent, nitrosamines, and others. So all can be used or all can be have a extractable leachable category. Okay. So for example, I have selected phthalates like PET bottle. We are generally said the PET bottles. So PET bottles is what? Phthalates, polyethylene tetrathalate. So for making of phthalates, there are several categories there: dimethyl phthalate, diethyl phthalate, dibutyl phthalate, diethyl exyl phthalate. So these are the few of the examples which are comes under the category of phthalates, and which are used for making of the plastic as a plasticizer. Okay, to make a plastic more durable, more flexible. So these are the compounds can be are already used in the pharma for making of plastics. So again, so why we have to concern about these phthalates? It has a several severe potential side effect like endocrine disruptor, and major side effect is male infertility. It will greatly affect the fertility of the male and decrease the sperm count. Anti-androgenic properties. It will also irritate the lungs. It have a genotoxic effects, and some of the compounds like DEHP diethyl exyl phthalate are suspected to the carcinogen. Okay, so amongst that, all the phthalate categories, dibutyl phthalate and DEHP diethyl exyl phthalates are most toxic among phthalates. So they produce the infertility and they produce the carcinogen. They are also considered as a carcinogenic substance. So we have to very much clear about that. These are the very good. These are most important things to analyze these things in our daily consuming products. So several regulatory guidelines are also there for pharmaceutical packaging. So in US and Europe, we can say it is a more stringent guidelines regarding the use of phthalates. So several countries has already banned the phthalate material for use in the plastics because of their side effects. Okay. So in USP chapter 381, USP 660, 661, 1663, assessment of extractable, 1664 assessment of leachables, European guideline also, EMA guideline and ICH guidelines also as well there to give the some highlights that how to what are the qualities of the packaging materials and what the limits are there and what are the what things which are not present in the product. Okay, so these are the several guidelines, but unfortunately we have in India we do not have any strict guidelines in a PET container, but in Europe and European US market it is stringent guideline. So I'm talking about the some kind of literature survey. So because we are discussing about the HPTLC, so if you find the extractable leachables in GC or GC-MS, there are several number of methods are available in GC-MS, because this GC-MS is itself considered as a sensitive techniques for the and for the extractable leachable are the trace elements or trace contaminants. So it is better suitable for GC-MS. Then LC-MS, you can find the numbers of articles on toys, food, consumer and cosmetics, medical device, pharmaceuticals and waters. So if you find the extractable leachable with GC-MS or LC-MS, there are numbers of methods are available. But in case of HPTLC, there are no methods available or very limited research are ongoing or very limited research are there for the HPTLC. So we thought that we have to establish the extractable leachable study by HPTLC method, and we have started our this research in 2015 and 2018, 2015 to 2018 we have done this research, and we have filed the patent also. So in HPTLC, we have directly come to the experimental work. So we have selected the four phthalates, and the four phthalates are structurally identical. Only the one functional group is different, so it is difficult to separate. But we are succeed to separate these all the four plasticizer, or we can say phthalates, which are generally used for making of plastics, and they can be leached into the product, and they will produce the harmful effect in our body. Okay, so these are the chromatogram of HPTLC, and these are the structures of phthalates which I have used. These are the method specification. We are getting the very good methods and rapid methods with a good separation of RF. Sample selection, because few of the questions are already there in the form that how you have prepared the samples. So I have included the sample preparations and sample selections. So sample selections means in parenterals, or we can say the probability of finding the leaching or extracting material, it is high in a parenteral preparation rather than the capsule or tablet, because in that the product is directly not much in contact with the product. But in parenteral, we can say the liquid samples or whatever the injectables that are directly injected into our body, and the surface area is also large. And most in India or in all over the world, the parenteral preparations are, or large volume parenterals are mainly packed with the plastic container or PET bottles. So these PET bottles, or the plastic of these PET bottles, were easily leached and into the found in the product. So we have selected the parenteral preparations. All our parenteral preparations we have selected because the leaching probability are more in that these. Okay, and we have procured from the market several formulation we have procured from the market, which have packed in the plastic container and which is also considered as a parenteral preparations and packed with the plastic. So sample preparations for leachable study. So we have selected the bottles, and 30 ml of product we have taken. We have add because we have extracted with NXN. So NXN we have to add and we have extracted for some times, and the organic layer is collected, and we have evaporated and reconstituted 1 ml to the methanol, and we have applied into the HPTLC plate. Okay. So this is for leachables. So for leachables, we have to take the product as for the extractions, and for extractable, the remaining protocols are same, but we have to just apart from product, we have to cut out the plastics, and the plastics we are need to going to extract, uh, and then that extract will further followed as per the same procedures for the leachable studies. So this is the sample preparation protocol. Some of you have asked in the questions also. So these are the sample preparation protocol in the extractable leachable study in our case. So result and discussion. So I have highlighted directly the slides which have a standard as well as test. So you will find the there is a DEP, means 4 phthalates are there in left side. There are 4 samples are there, and 4 samples are properly separated. In right side, it is a sample size, a sample application is there, test sample. So we have extracted. So we have found the so many things from this experiment. So this is the method validation summary. My method is compatible with 100 nanograms, so which is good enough for extractable leachable studies. Okay, so 100 nanograms I have gone to up to the 100 nanograms, so it can be detect up to 100 nanograms. Sample analysis. After sample analysis, as I said, there are 13 samples we have selected. So after doing the scannings, we have found that 5 of the samples can be which is seen in the extractables, where 0 samples are from for the leachables, means there is a no samples, means there is a no DMP is found in the leachable studies. But in extractable, means in the forcefully extracting techniques, we have found the 5 samples with the DMP concentration by HPTLC technique. Okay, and it is then our second analyte again. Uh, if we had there is a one sample is showing the response, and in leaching there is a 2 samples showing the response regarding the amount of the extractable leachables. Third, DBP, which is considered as the most toxic compounds amongst the phthalate category. So this compound has found in several samples. Okay, and as well as the DHP is the most suspected compound in the phthalate category, which is carcinogen and is also responsible for the infertility, making infertility in men. So we have found almost all the samples where in very significant amount of concentrations this component is found in almost in every samples, because DHP is the most widely used to prepare phthalates to make a more durable and more flexible. This component is mostly used, DEHP. So most of the plastic products are making from this content. So we have seen this content in every samples and in very significant amount of samples. So so it is obvious that this kind of studies are should be there. So in the regulatory forum or international forum, there are several standards are there, like class 1, class 2, class 3, class 4. Okay, and how much exposure of per day that is given by the limits, and this is threshold limits. So apart from our means after doing this study, we have concluded that DBP and DEHP, means diethyl phthalate, diethyl hexyl phthalate and dibutyl phthalates, were dominantly present into the pharmaceutical product, which is plastic packed and is parenteral preparations, and parenteral preparation directly injected into the blood. So you can imagine that what kind of side effects can has been done by these components, and which was considered as a more toxic, and which is fall in the class 1 category. So whereas DMP or 2 analytes are under the threshold limits, but uh, for 2 components it is a more toxic. Okay, so this is about the pharmaceutical estimation of pharmaceutical product uh, of extractable leachable. So for this research, I have filed the patent, and it is also published in the 2020. Okay, so means we thought that the method cannot be patentable, but after 2019, there is a amendment in the regulations of patent filing. The method can be also patentable. So previously it was not there, but right now it is there. And China is very smart in this sense. China is taking the patents on each and every methods, because we are doing lots of work in method development and validations, but we are not aware about the patents. Okay, but China is very smart. Most many patents are found in HPTLC, LC-MS, HPLC, means develop the methods and filing for the patents. Okay, so similarly, the trends has also been started in India also. So for as a part of that, I have also filed the patent, and it is also published. So it is very good that HPTLC methods are also in the patent list in the India. Okay, so as a conclusion of this work, that pharmaceutical means extractable leachables is the main concern as a packaging material, and which is present in pharmaceutical product. So we thought that, so this is a 1 year exercise. So we thought that, so because the plastic is not only for the pharmaceutical product, the plastic is everywhere, like in waters, we have food, waters, like cosmetic we are using milk. So everywhere, suppose the room sitting inside and which is plastic paint, so we are also surrounded with the plastic paints wall, and the fumes of that plastic is also inhaled by our body. So means we can say that the exposure can't be controlled, because plastic is everywhere. Suppose the water, so in our house previously it was a concrete well, but now there is a plastic bottles above our terrace or in our apartments. Okay, so previously it was concrete well. So we cannot avoid plastic right now. So plastic is everywhere, means plastic we can't avoid. So we cannot say that which is the source of phthalates, which is the source of extractable leachables, because it can be comes from anywhere. So we thought that, okay, just checking into the pharmaceutical product, it is okay, but the pharmaceutical product is taken by the patients, that will be gets metabolized, that will be also identified. So we have also done some research and literature review, and we have found that the plastic is everywhere, and plastic is many ways plastic ingested in our body. But all plastics will hydrolyze, means the extractable leachables and particularly phthalates category, they will hydrolyze, and it will excrete more than 90% of in urine. So we have thought that why not we check the urine samples, because the plastic can be come from any side, from any ways. So, but it will excrete through urine more than 95%. Okay, so we have thought that we have to also check that phthalate metabolites, because the phthalates are going to hydrolyze. The phthalates means it is diester phthalates. I will explain over here. So this is the, so this is the phthalates basic moiety, diester phthalates. So the R group will determine the which kind of phthalates is there. If it is methyl, so both end is methyl, so it is called as a dimethyl phthalate. If it is ethyl, so diethyl phthalate. So it is like this. So it is parent basic moiety, and once it is exposed in our body, so it gets hydrolyzed by enzyme, and it will convert it into monomethyl. So one group is removed over here, so this R will remove uh, here, and it will create the dimethyl phthalate to monomethyl phthalate, dibutyl phthalate to monobutyl phthalates, and likewise. Okay, so these are the uh, compounds which will used in our study. Like in pharmaceutical product, we have used the product which is you mainly use for the packing preparations. But when we have analyzed the urine samples, then we have used the metabolites. So this is for your information. Then the phthalates are diesters. Once it gets or in our body, it will metabolize to monomethyl, and the dimethyl will gets converted into the monomethyls. Likewise, the other compounds are also converted into the monoesters. So this is the compounds which we have selected for the biomonitoring, because it is the metabolites, and these are the this can be found if the body is exposed from the plastic which have made up of these compounds, dimethyl, dibutyl, dibenzyl, and DHP. So they will get metabolites in our body, and they will extracted through urine with this. So these are the sample preparations. We have procured the standards from the Sigma L Reach, and we have prepared the standards 1 milligram per ml. And we have to also identify from the urine sample. So these metabolites are generally conjugated in the urines. So we need to do some enzymatic reactions for the deconjugations. So we have used the beta glucuronidase enzyme for the deconjugation of these metabolites. And we have prepared 200 units per ml sample for these reactions. So also the extraction process which we have followed. So we have taken 1 ml of urine samples, and we have spiked with our metabolites. Then we have spiked the enzymes for the deconjugations. We have incubated, extracted with 2 ml ethyl acetate, and each steps are optimized. Suppose we have said what is the effect of beta glucuronidase deconjugations. So we have extracted with without extractions, without enzyme, and with enzyme. So without enzyme, the recovery is more nearly about 50%. But with enzyme, you can see the average of 41% of the recovery is increase. So we have finalized that with enzyme or without enzyme, there is a significant difference. So we have used this enzyme for the deconjugations, and we have received the significant increase in the recovery. Similarly, ethyl acetate. So we have tried with the several extraction solvents like DCM, TBME, diethyl ether, ethyl acetate, and hexane. And we have found that the recovery in terms of ethyl acetate is more. So we have, and it is consistent. So we have used the ethyl acetate for the extraction process. Okay. So this is the photographs and chromatogram of HPTLC. These are more similar, structurally similar compounds because these are all are mono esters. So these all are structurally similar compounds. It is difficult to separate, but even though we have success to separate the all four compound with good RF. Okay. So this is our chromatogram and photographic photo documentation of this development plate, TLC plate. This is our methods. We have used the within 15 minutes we have completed this analysis. So this is very much suitable. So you can see the RF value. RF value is consistent 0.4, 0.5, 0.6 or 0.7. So it is also good enough. The validations, you can see the all parameters, because it is a bioanalytical method. It is not a simple method, it is a bioanalytical method. So in bioanalytical methods, we have received the good linearity 0.99 in all the cases. This is the bioanalytical method validation summary, that linearity, precision, recovery, matrix effect, and stability studies all are within the limits and as per the US FDA and whatever the ICH bioanalytical guidelines are there. So all are within the limits and as per the guidelines. So there is no any issue related to the reliability of the method data. It is equally reliable as the other techniques. Then, as I said, we have developed the bioanalytical methods, and then we have made hypotheses that which patients are more prone to have this kind of metabolites in their urine. So we have made two hypotheses. That one patient, which is because earlier research indicate that parenteral preparations having a more amount of extractable leachables. So we thought that ICU patients can be taken. So in ICU, mostly parenteral preparation dose is administered in the directly blood. So because we have already found that in parenteral preparations there is some traces of the phthalate component. So we have taken the ICU patients where we have showed about the uh, in because the parenteral preparation is given administered. So we have sure about that plastic exposure is there, and we have second hypothesis we made that fertility centers, because these phthalates metabolites have many major side effects of infertility, because previously in metro cities the fertility centers are there, but right now small towns and districts have also fertility centers. So there are rapid rising in the fertility clinics, and there are more patients, because we can say that we have collected the samples within a week. Okay, so we can say that how many patients which are undergone the treatment of fertility. So we have made a second hypothesis that infertility patients can be taken, because the one of the side effect is the infertility. So in Rajkot we have made this exercise. So in one hospital, one we have selected the 14 samples uh, uh, from the ICU patients, and two fertility clinics. We have made 31 kind of or 37 kind of samples from the. There is a patient. I am talking about the patient. Okay, so 51 sample is already under the our in our study. So you can find this is the standard TLC plate, standard applications, and these are the urine extraction of urine samples. You can find that many of the samples, many of the metabolites, which is found in the patient urine, and which is in a significant amount. So we are also surprised that how much they are going to expose in our routine sites. So, uh, so you can see the patients or the subject which are more relevant to uh, uh, this leaching probability, or they are more relevant to the finding of these samples from the urine. So they will have a more amount of sample is there in their urine sample. So after calculations, we have done that. We have selected four metabolites. So different patients have a different concentration of samples in their uh, urine. But we just look at the graph that the metabolites are found in their urine samples, and these metabolites are, or these compounds are having a severe side effects, and which is our in our daily, because we don't know that because we are all are using the plastic samples, plastic preparations in our daily, like milk, food, water. We don't know what is the source of contamination, what is the source of in our body. But in urine samples, the concentration of these phthalate metabolites are significantly found. If we further categorize uh, like phthalates, lowest value of these metabolites in any samples out of 15, it is 0.8 to 47. You can imagine 47 microgram. Okay, and likewise in methyl benzyl phthalates 0.1 to 11, monobutyl phthalates 0.3 to 2, and methyl ethyl phthalate 0.2 to 32. So you will surprise that how much quantity of plastics are going to expose in every day of our life, and what kind of things are there. We have taken the samples of patients. So in patients, these are the quantities are means these are the quantities are available in their urines. So we don't know what is the plastic exposure in our body, but if we want to analyze what is the plastic exposure in our body, so we need to check our urine samples also. So if you are further categorize the patient number of patients, so in 0 to 10 microgram per ml, almost most of the patients are relying on the microgram per ml. So we can say more or less the every patients are getting exposed with the plastics with a small number of phthalates metabolites in their urine, and very few of that patients like which is fall in the very severe category, which is 40 to 50, 30 to 40, and likewise 110 to 20. So we can say that this type of study is very needed in the our Indian populations, because we are having a rising in the cancer patients, we are rising in the infertilities issues. So and we are treating with the medicines, don't changing the lifestyles. But after this, we have advised our doctors who have participated in these studies. So and they have advised that you have to minimize the plastic contents from your daily routine life. So it is one of the now prescriptions, or prescription means one kind of the uh, uh, we can instructions given by the doctors. The patient having infertility or patient having cancers, there are after these studies the doctors are giving the instructions, because in Rajkot we have also highlighted in the media like these studies and the effect of plastics. So after these, the in the Gujarat or particularly in Saurashtra regions, the doctors are also instructing the patients that you need to avoid the plastic exposure as much as possible uh, to your daily routine life. Okay, so this is all about the biomonitoring of phthalates, because first we developed the bioanalytical method, and we have applied to the patient sample, and we have monitored the concentration of phthalate metabolites in the human samples, okay, human urine samples. And we have also, because this is also first time we have approached the metabolite study of extractable leachables in by HPTLC method. So we have also filed this patent, and it gets also published in 2020. Okay, so we are also happy with the method filing and method. Our methods is if our method is suitable for the biomonitoring, then we are more happy, because these studies very much needed in human populations, in Indian population, like, because we are not concerned with, because in US or Europe market it is highly regulated markets. The in several countries PET bottles are banned. In looking to the I'm just now quite different, quite deviating from my topic. Like in 2014, because where we get the idea about this study, because in 2014 the CDSCO has published CDSCO has published one report. The report name is YK Gupta Committee report. In report, they have mentioned that PET bottles, PET containers in pharmaceutical products. So they have discussed about that several countries has already banned the PET bottles for pharmaceutical products. So what situation in India? So they have formed the YK Gupta committee, and they have filed the reports, and that report is available on CDSCO website. That in 2014 December or 14 December or 15 December, the website report is there. So he has suggested that in India is a very much widespread continent, and we cannot assure that where is the contaminations or where is the source of contaminations. But the laboratories, governments, universities, and academics will need to start work on it, and provide the good database and provide the good data on these things. So the government can draft the rules. The government can draft the uh, any regulatory guidance documents will proceed. Okay, so we have thought that we have to start these phthalate means PET containers, particularly phthalate contents in the pharmaceutical product as well as from the biological materials. And my third part is the comparison of with method with LC-MS. So because this topic, extractable leachable, is not topic new for us, because we are since working in 2013 in extractable leachables, and because the LC-MS is a good choice for the extractable leachables. So we have from 2013 to 2015 we have worked on the extractable leachable study with LC-MS. But while doing the LC-MS, we feel like, because what water we are using as a mobile phase, what methanol we are using as a mobile phase, what solvent we are filling into the HPLC system, they all having a plastic container one or other place. So we cannot say that where is coming from, because uh, water is also filled in the plastic container, because if you have a millipore system in your lab, so that water, HPLC grade water, is also there in a plastic container. If you are procuring the HPLC grade water from the market, it is also filled in the water with plastic container bottle. So the concentration is already, some of the concentration is already there in the blank also. So it is difficult to achieve the sensitivity, or you can see the baseline in LCMS. If you are experienced with the LCMS, then you will have a very good understanding regarding the baseline and mass saturations. Okay, so you can find that in graph that baseline is quite high. It is very much high baseline, because the reconstituent solvent and as also solvent we use, because the solvent have also some of the compound, because it's also exposed some of the time with the plastics, so it has also some of the component in there, and it will detected by the instruments. And these are our standards in LCMS. But I'm just talking this graph. This is the baseline saturations. So common practical hurdles with LCMS, because we have also developed one of the paper is also published in the very good journal like Extractable Leachable study by LCMS. So I am comparing this my method with my this HPTLC method. So like as I said, baseline saturations, or we can say mass saturations, because this component is find like DMP, DEP, DBP, and one of the compound DEHP, which is, as I said in HPTLC analysis, it is also said that it is mostly found in all the containers. So DEHP is mostly used in the plastic container bottles. So it is constantly running into the mass. Okay, so constantly running into the mass, so it will give the baseline saturations or mass saturations. So you cannot achieve the sensitivity. You cannot achieve the proper analysis in compared to the in simultaneous analysis with the others. So we have excluded the DEHP in this LCMS study. But now I am comparing of my method, HPTLC method, to LCMS method. So if we talk about the sensitivity, so obvious LCMS I have reached up to 1 nanogram per ml. So this is most important part. This is those who are working with HPTLC. So because there is always question on mind that why not LCMS and why not HPTLC. So I have fully aware about this. What is the benefit of LCMS? And I am not said that LCMS is not replaceable, or LCMS is not a good choice. LCMS is also a good choice. I am also working on the LCMS, so it is also a good choice. But HPTLC can also be an option or good option for this kind of study, like sensitivity. So in LCMS, I have reached up to 1 nanogram per ml, but in HPTLC, it is 100 nanogram is also enough sensitive. 100 nanogram per band, it is enough sensitive for the extractable leachable study. The number of phthalate analyzed in my HPTLC method, I have analyzed 4 phthalates. Like, but in LCMS due to mass saturation issue, I am able to analyze only 3 samples. Okay, method developments in HPTLC, it's quite simple. You already all know. In LCMS, it is quite complex because you have to look at the more parameters. Method validations in both, we are able to comply all the validation parameter in kind of ICHQ2 guidelines, or we can say bioanalytical guidelines. Sample analysis, in both cases they are quantified, so there is no issue regarding the quantifications. Sample processing, we are quite flexible or quite relaxed when we are sample preparing the HPTLC rather than the LCMS. So sample preparation is simple, while in case of LC-MS, sample preparation is quite complex, and we have to more concern while we are preparing the samples for the LC-MS. Instrumentations simple. In case of LC-MS, it is quite complex. We need to look all the parameters. Cost wise, as well as you already know, the cost wise HPTLC is quite economical than the LC-MS. So I have covered the all. I have found that all these things are very much useful than the LC-MS. So I thought that why not file for the patent, because all the parameters, all the functions are giving me the that it is beneficial toward the existing. Okay. So what is the benefits? Or there is a baseline mass saturations can be avoided, or it will be there in LC-MS. Complex sample preference there, more number of optimizations, complex instrumentations, and greater cost. That can be, we can say for the LC-MS, it is by default. For any analysis, it is by default. Okay. For HPTLC, I have compared with my in-house method to HPTLC and other techniques, because we need to discuss the LC-MS because we have discussed, but what about other techniques? So we have compared by application wise and cost wise. So in application wise, the LCUV can be applied for the chromophoric compounds. GCMS can be for the volatile and semi-organics. LCMS for non-volatiles organics, and ICP-MS for elementals. And also for cost wise, LCMS it is 8x, means more costly than HPTLC. ICP-MS it is 4 times costly, LCMS is 4 times, GCMS is 2 times, and LCMS simple HPLC is equivalent. So if I put now HPTLC or TLC-MS, so it have all kind of applications. It is fit for purpose for all kind of application, we can say. And the cost, it is nearly for the HPLC with same benefits of LC-MS. The HPTLC can be used in our analysis. So it is my personal experience that I have replaced the LC-MS for this study, and I have started with the HPTLC. It is because of the these are the advantages. Okay, so it is a cost-wise analysis and application-wise analysis. So as a part of conclusions, uh, the HPTLC is the essential instrumentations, and it is fit for purpose solution meeting regulatory aims without over engineering. So in LCMS there is over engineering is there. So in HPTLC there are simple instruments and it is fit for purpose. It may be useful for the routine quality control tool which significantly minimize the cost. If extractable leachable analysis with LCMS, then you need a very specific setup. While the same setup for HPTLC you can use for the extractable leachables. If we talk about the sensitivity, so sufficient analytical evaluation threshold has been established suitable for safety concern threshold. So whatever the safety concern threshold is there, it is already achieved with the HPTLC techniques. So no need to going more sensitive and more sensitive part like LCMS. It is okay, like you have to found the like analytical thresholds, like it is more sensitive or more advantage on methods. But when we talk about the safety concerns, so whatever the safety concern threshold is there, it is already achieved by it is achieved by the HPTLC methods. So you can also use for the bioanalytical and biomonitoring of these metabolites. Okay, and HPTLC can also be effectively utilized for the biomonitoring of phthalate metabolites from the humans. So I have covered the two parts. One, the extractable leachable study by HPTLC techniques from the pharmaceutical product. The analytes which I have selected, which is mainly used for the making of the plastics to make a more flexible, durable, and the phthalates are used as a plasticizer. And then I have extended my study to the biomonitoring level, that I have developed the method from the urine samples, bioanalytical methods, because the plastic exposures can be come in anywhere. So, but it extract in a common pathway, more than 95% will be extracted through urine. So I have tested the urine samples, and I have also found the phthalate metabolites in the urine samples in the uh, more prone patients like ICU patients and infertility patients, okay, which are the main side effects of the phthalates metabolites. So we can correlate. I'm not saying that this infertility is due to only phthalates, but I can correlate because it is already reported, and this infertility is irreversible. I'm just talking about it. This the phthalate causing the infertility, which is irreversible. So you cannot find the study on human, okay, because the study which is not feasible in human, that you cannot find in the human. So irreversible reproducibility, irreversible infertility is causing. So it cannot be cured later on. So this kind of study you will not found in human, but you will find the reports on animals like rat and rabbits that have a infertility issue. They are giving the exposure of phthalates through the toxic levels, and they are causing the infertility, and that cannot be treated normally, or that so it is thought that it is reversibly irreversible. So we have to more concerned about this, the use of the plastics and this. So this study indicates two things. One is the use of HPTLC in a perfect way, an efficient way, and second things, to avoid the plastics in daily routine life, to avoid the major side effects of these phthalates. So with this, I will acknowledge my university, my previous university, Soras University, my current university, Gujarat Technological University, Encrom, and also I'm also thankful to Chemeg, which have made the very nice instruments, and also thankful to Encrom to given me opportunity, because I have also taught these techniques at several international platforms in like in HPTLC in 2017 in Berlin, and consecutively in 2018 I have also discussed this technique in Sydney, Australia. And so I have, whenever I get the opportunity to explore the HPTLC technique, so I have raised this concern regarding the use of the minimization of use of plastics and use of the HPTLC. So these are the two messages from the takeaway from these presentations. The HPTLC is a good choice for these kind of applications, and to avoid or to minimize the use of plastics of your daily routine life. So with this, thank you for your kind attentions. Uh, I'm very much thankful to all the audience who have passionately listening throughout the sessions, and also thankful to again Encrom and Saikat ji sir, Ramesh Naidu sir, Akshay ji sir for giving me the opportunity. Over to you, sir.
Yeah. Thank you, sir. You know, it is a wonderful session which we had, because you see the application, instrumentation is one part, and using the instrumentation for a noble cause is a different part. Here we see the second application very wonderfully mixed in with the utility part. And so, as I told you, sir has been very dynamic, and this represents that how interestingly he has presented both the concepts. By it is a bioremediation kind of topic. You can correlate to the environmental sector. You can correlate to a packaging material industry, at the same time food industry. So it has a huge dynamism of the topic, which can be correlated to any field, especially phthalates. We know these poisons are there, but their exploration has been very limited. But now, with such kind of a research, it has been very beautifully highlighted how it enters into the food chain, or the human life, and how they are getting problematic for us. So thank you, sir, for sharing this wonderful presentation and this insight. And the takeaway which sir has given is very precisely concise in a very short capsule way. So thank you so much. I think this is one of the best lectures which we have in the series which we have. And thank you so much. Our people have definitely benefited, especially the researcher group which is there present over here. Now, before we summarize and end this session from our side, we'll take a very few questions which are very pertinent. As you know, this is a patented technology, so certain things will not be revealed to as per the jurisdiction of that patent rights. So only if sir wishes, we will be providing you those answers. Otherwise, you will have to contact him personally and get the details from him. He is very open and he is accessible to any person who wants help. So that kind of a person he is. So you don't feel anything. You can definitely write to us and sir and get the details. And thank you once again to this fantastic, definitely you work over there, but the facility which is there, that should be also compatible. So special thanks to his department, Gujarat Technical University and Saurashtra University, for supporting such dynamic researcher who has changed this field of lookout and interesting subject which he has highlighted and pointed out to us with a very beautiful presentation. So now we'll quickly take up few questions and we'll see whether they come into the purview. So first is relating to a method. There is a question by Mehul Mehta who has asked, ethyl layer was taken or an aqueous layer was taken for sample preparation?
Uh, First of all, ether is not there. It is
Hmm.
ethyl acetate.
Hmm.
It is ethyl acetate. So ethyl acetate layer was taken,
Okay.
and it is evaporated and then reconstituted with methanol. So
Okay. Okay.
we have extracted with ethyl acetate.
Thank you.
We have tried with the many solvents, like I have said in my presentations
Hmm.
also.
Hmm.
I have extracted with diethyl ether. I have extracted with TBME, tributyl methyl ether, ethyl acetate, hexane. Many solvents I have used, and I have compared my recovery.
Hmm.
So I have found the ethyl acetate is a good solvent for my recovery. So ethyl acetate I have taken from the and it is evaporated.
Okay.
Then it is reconstituted with 1 ml of methanol.
Thank you. Thank you, sir. Second question is also a general one. The person has asked, leachable studies, is it streamlined only to plastics?
Uh, yeah, more we can say it is not only because of plastics, because the sometimes you will find also some articles on glass containers. So it is a plastic is a very most significant category, but the other category is glass, and a few other things are also there. Glass and plastic. Generally, we are finding the two containers, plastic and glass.
Right.
So if we talk about the containers, so these are the two categories. But if we talk about the share of leachable studies, so hardly 5 to 10% of the studies which is covering the glass container,
Glass
but
content.
more than 90 to 95 studies are covering for the plastic containers. So apart from plastic, glass container is also there. So we can also do the see. And if you are talk about in the selection of the research topic from the containers, so whatever the container is there, you select the what is the content which is used for making up this container, and you analyze that which are if there is a side effect or not. If it is side effect, then you can made a hypothesis that it can be present in our formula in product, and you can start your analysis as a leachable or extractable study. But for uh, category of studies, there are two containers, plastic and glass. So these categories are there.
Thank you, sir. Uh, we'll take another two, three questions last, and then we'll sum up the session. One question is, why beta glucosidase and not any other enzyme was chosen? Of course, sir, if you feel the question is infringing the rights, you can not answer those, and you can ask us that the answer will be sent through mail. Okay, sir.
Okay.
So I
Not,
just, yeah.
not an issue, because the person who are uh, I personally feel that science is open for all. Okay, so someone is really interested and working on the same topics, then I would be more happy. Uh, these enzyme generally used for the deconjugation of metabolites. So particularly we have selected these enzymes. There is a no we have a list of enzyme, and we have then selected particular this one. But this enzyme is generally used for the deconjugations of analytes from the urine. So that's why we have selected. There is no any other rationales, or there is no we have a list of enzymes, and out of that enzyme list we have selected any one for particular reasons. But generally, we have tried with without the enzyme, because if you have seen my presentations, we have tried with without enzyme and with enzyme. So you can try with any enzyme. It is not a literature. You have to not always rely on a literature. You have to do by your own. You have to first select any of the enzyme which is known for their deconjugation, or any fit for purpose. You have to do without and with, and you need to compare both the things, and you can check the what is my improvement. Okay, and this improvement is satisfactory to you, then you can opt that options. Otherwise, you can go for the another deconjugation enzyme, and you can again try with another enzyme. Okay, but you have to prove that why, what is the benefit of using this enzyme, why you have to use this enzyme. Apart from that, uh, what is the effect of this enzyme, that part is very much important.
Wow. Thank you, sir. And then people have asked like any specific specifications of sonication and all those things you will find in the paper which sir has published. We'll not go into discussing those things. Can you share some references of presentation? Of course, we'll ask the permission of the sir, and then we will make it available. Now we'll take up one question. Somebody has raised an hand. We will like to unmute them. Your marketing team, may I request you to just give the permission to the,
Yes, sir.
yeah. Yeah, ma'am, we have authorized you to unmute your mic and ask the question. Venumula ma'am, Padmavati ma'am, if you can hear me, we have given you the rights. You can unmute yourself and ask the question. Padmavati ma'am, hello. Can you hear us? If you can hear us, we have given you the rights. Just unmute yourself and ask the question. Till madam figures it out, we'll just take another question. This is in the chat box. Majority of the questions sir has already covered. I'll just take up which sir has not taken in. Okay. Sir, can we file the patent of HPTLC method in herbal drugs? That is, some person has asked. So anything which you can, sir, throw a light?
Yes, yes. But you need to first do prior art search for patent. So if you find that your prior art search is very good enough, and you have a strong determination that you have these methods or this work has been not reported with the HPTLC method, and if the methods are already available with the other techniques, then you need to also highlight the, you need to also claim the what are the benefits with your methods. Okay, so these two things are there. Prior art search, you need to do the prior art search, where you can find that whether there is there any method is available or not. And not only for HPTLC, you have to also look at the other methods, and you have to find the benefits of your methods, and accordingly you need to claim those benefits in your uh, patent claims. So these 2 to 3 things you need to concern when you are filing for the patents, because I have also received the grant for filing the patents for these 2 topics. So I have hired the patent agent, and they have done the private art search. So they have done the professionally private art search, and they have found that there is no work on that, and that can be a good candidate for the patent applications. So for that, after that, these things will be there.
Wow. Thank you, sir. And rest all questions we have also covered in. So any of more questions, please feel free to write at lab@ancrum.in. If you have any questions directly written to sir, you can send it to us. We will forward it to sir and get the answers back. With this note, we'll conclude this session. And first of all, our heartiest we extend our heartiest uh, uh, thank you, sir, for this wonderful session. It covered everything. It was a joint package of the best side of HPTLC and other facets of a research work. So thanks, sir. I am running short of words, as you can see. So thank you so much once again for educating and sharing your knowledge on this platform with all the young research minds. So with this, again, on behalf of Akshay sir and entire Ancrom team, we extend a special thanks to you, sir. And with this note, we would like now to end this session. So with your permission, we would like to end this session, sir.
Yes. Again, I am thank you. I wish to thank you all. I am also thankful from my whole GTU team, on my behalf of GTU and my own GSP team. And I'm also thankful to the all the Encrom team who have supported anytime, because we have also the full-fledged instruments of HPTLC and many applications they have developing, and many times we have contacting you. So for this, I'm also thankful to all the team of entire Encrom team. I'm also thankful to all the participants who have passionately listening these all the sessions, and I'm also thankful to the Almighty God. Thank you all.
Thank you, sir. Thank you, sir. Sir, with your permission, we
Yes,
will like to end the
we
session.
can we
Yeah.
can